Hello again, anatomy students. In this lecture, we will be going over the lymphatic system. Be sure to refer to your syllabus for specific due dates. You have, or should I say, have had about 8 weeks to complete this lab, so you should be, or should have been, going to open hours or using free time in labs to look at the lymphatic slides and models. The test for this lecture is online and is listed under the Lymphatic System tab on your BB Learn shell. If you have any questions, make sure to ask your TA. The discovery of lymph nodes goes back to Hippocrates in the 5th century. As we'll soon see, the lymphatic system is a very low pressure system. So most of the vessels collapse upon death, which makes finding them extremely difficult. Because the nodes were filled with a clear fluid, the term lymph was derived from the Greek word nymph, meaning a creature associated with clear streams, and the Roman deity Lympha, meaning clear spring water. It wasn't until the 1600s that many of the lymphatic structures, like lacteals and the cisterna chyli, were discovered, and the story of the lymphatic system started to come together. What's always great about science is that we know a lot, but there is always more to learn. So keep your curiosity fueled. You never know when you will make the next greatest discovery. What should you learn from this lecture? Well, first, you should walk away with a good understanding of the function of the lymphatic system as a whole. Second, you should be able to identify lymphatic organs in the body by using images and models. Once you have that down, you will need to know the functions of those organs. After that, you will need to describe the movement of lymph through these organs and throughout this system. And as always, you will be required to identify organs and structures under the microscope and make representative drawings in your notebooks. The main functions of the lymphatic system are to recover lost fluid. As you sit and watch this video, your capillaries are leaking and this system works to recover that lost fluid. Don't worry, you don't need to call a plumber. This helps your immune system monitor for any pathogens. And this system works together with your digestive system, absorbing lipids from the small intestine. The title of this slide is hyperlinked to some helpful Khan Academy resources. First, let's go over some terminology. Lymph is the recovered fluid this system is responsible for picking up. Just like blood vessels transport blood, lymphatic vessels transport lymph. To the right is what a lymphatic capillary looks like. Maybe you notice something a bit different about its structure. Tissue is made of cells, so lymphatic tissues are aggregations of cells like lymphocytes and macrophages. And lymphatic organs are concentrated lymphatic tissue, so a lymph node would be one example. Let's take a closer look at the lymphatic capillaries because this is where all the magic happens. First, let me orient you to this image. Here's a blood capillary. Surrounding the capillaries is connective tissue and cells. And this large green structure in the middle represents the blunt-ended lymph capillaries. As the circulatory system is transporting blood to the blood capillaries, oxygen and other nutrients are diffusing out and into the surrounding tissues, while CO2 and other wastes are diffusing in. While this is happening, blood plasma is also leaking out of the blood capillaries into the interstitial space. Once it leaves the blood capillaries and enters the interstitial space, it is referred to as interstitial fluid. The interstitial fluid enters the lymphatic capillaries because the pressure in the interstitial space is higher than it is in the lymphatic capillaries. Therefore, the fluid moves along the concentration gradient. Once it enters the lymph capillaries, it is referred to as lymph, and now it travels through lymphatic vessels to lymph nodes to be cleaned and monitored, and it will finally be dumped back into the blood circulatory system. You can think of this system as an internal bath for your body. That interstitial fluid is going to pick up waste, debris, or pathogens and bring it into the lymphatic circulation. Anything foreign will get removed or initiate an immune response. Here's an image from your textbook. To be clear, you will not need to know the specific names of pressures or any numbers for lab. Your lecture professor will most likely go over this diagram in detail, but this shows you why plasma leaks out of the blood capillaries. To orient you in this diagram, we have a close-up view of a blood capillary with blood flowing from left to right, as illustrated by this arrow. We also have a lymph capillary up here and some cells from the surrounding tissue. Because this is a pressure-driven system, you can see that on the arterial end of the capillary, all those different pressures listed in the yellow box cause a net filtration out of the blood capillary. On the venous end of the blood capillary, all those same pressures create a net reabsorption into the blood capillary. There's a lot going on here, but the take-home point, especially for lab, is here in this red box. Notice on the arterial end, there is a net filtration of 10 millimeters of mercury, while on the venous end, there is a net reabsorption of 9 millimeters of mercury. The negative represents the direction of the fluid moving out of the blood capillary. So, when you compare these numbers, there is slightly more filtration occurring than there is reabsorption. So think about it like a clogged drain, which I'm sure many of you as college students have right now. But when the amount of water coming out of the faucet equals the amount of water going down the drain, there's no problem. But when more water comes out of the faucet than can go down the drain, fluid starts to accumulate in the sink. That is the interstitial fluid, and it does provide a lot of benefits to your body, not to your sink or your bathroom floor. You can also think of the lymphatic system as that little hole near the top of the sink rim that reabsorbs the extra fluid. If too much fluid is coming out or not enough is being reabsorbed, that's when swelling occurs, or in this analogy, the sink overflows. Now that we know how lymph is formed, let's discuss a bit about its contents. Remember, lymph was originally blood plasma that passed through the interstitial space and then into the lymph capillaries. The composition of blood plasma varies based on our diets and lifestyles, as does the interstitial space depending on the body region. Because of this, the composition of lymph can vary. So in general, we will find different levels of the following. Lymph contains metabolic wastes and cellular debris, and pathogens like bacteria or viruses. Remember, lymph is essentially bathing all the surrounding tissues and taking the debris back to the lymph nodes, which will scan and clean the fluid. Lymph also contains lipoproteins called chylomicrons responsible for transporting fats. This happens through lacteals in the small intestine. It will also contain nutrients, and lymphocytes are B and T cells. So we've learned how lymph is formed, how it moves into the lymph capillaries, and what it's made of. Now we'll follow the path it takes on its way back to the blood. The lymph capillaries are all over our body. You can see an image of what they look like on the right side of your screen. One of the major differences between lymph capillaries and blood capillaries is that lymph capillaries are blunt-ended and not continuous. They have one-way valves, just like veins, and require compression through movement or from surrounding skeletal muscle contractions. Like we said in the circulatory lecture, exercise is extremely important in moving these fluids around the body. Every time you exercise, you are helping your immune system filter and scan the lymph to keep you healthy. This is a low-pressure system. Interstitial fluid needs a pressure gradient to move from the interstitial space into the lymph capillaries. The anchoring filaments hold the lymphatic capillaries open, and the endothelial cells overlap, which allows fluid to flow through the openings. But if pressure increases inside the lymph capillaries, it forces the endothelial cells closed so the fluid cannot go in the opposite direction. If the pressure in the lymph vessels does rise, usually due to some kind of blockage, maybe a collapsing vessel or a parasite, it's like clogging the drain, and interstitial fluid builds up in the interstitial space, causing swelling or edema. The lymph vessels have the same 3 layers that blood vessels do. Can you remember what they are? Tunica interna, tunica media, and tunica externa. The lymph vessels coming from the right side of the head, neck, shoulder, and arm lead to the right lymphatic duct, which dumps its contents into the right subclavian vein. The lymph vessels in the rest of the body leads to the thoracic duct, which dumps its contents into the left subclavian vein. This image shows you that division I just mentioned. The purple region is drained by the right lymphatic duct, and the rest of the body is drained by the thoracic duct. Here, you can see a closer view of what those 2 ducts look like and where they drain. In this close-up of the small intestines, we can see a lacteal inside of the villi. Villi create surface area to absorb nutrients from the chyme, the liquid substance that food gets turned into from digestion. Lipids are absorbed by the lacteals and packaged into chylomicrons, while other nutrients get absorbed into the surrounding capillaries to be sent to the liver. The organs you will need to study in models and under the microscope are the lymph nodes, the the spleen, the thymus, and the tonsils. There are hundreds of lymph nodes all over our bodies. You may have felt them in your neck when you are sick, or in the axillary or inguinal regions. These get swollen during an immune response due to the increase in immune cells and fluid. Each lymph node has a fibrous capsule, an outer cortex, and an inner medulla. As lymph enters the lymphatic capillaries and passes into the lymphatic vessels, it is transported to the lymph nodes via the afferent lymphatic vessels. Once inside the node, the lymph flows through the cortex and medulla past various immune cells. It leads through efferent vessels to another lymph node and repeats this process. This cleans the fluid and allows lymphocytes and macrophages to scan for anything foreign. You want to be sure to look at different slides of the lymph nodes before you take your online test. There are different types in the slide boxes, and they are all fair game. Remember, you are learning identification skills when using the microscope, and you need to be able to transfer these skills to any slide by identifying cells or tissue types, thinking about what their function is, and how it would help in the function of the organ you are looking at. Okay, so moving on. Let's go over the histology of a lymph node. There are two main regions which you already saw last slide, the cortex and the medulla. The cortex contains lymph nodules. To be clear, lymph nodules differ from lymph nodes in that they are small, localized collections of lymphoid tissue without a well-defined connective tissue capsule, and they are embedded in the reticular meshwork of the lymph node. In this lab, you'll also see lymph nodules in the tonsils and small intestines. These nodules have a germinal center that is mostly populated by B cells that will proliferate to make antibodies. The paracortex, a region deeper to the cortex, is mostly populated by T cells. As lymph passes through the cortex, it is exposed to these B and T cells, which are responsible for antibody-mediated and cell-mediated immunity, respectively. Next, the lymph flows through the medulla, which is mostly populated by macrophages. You should remember that these cells are large phagocytic cells that engulf foreign material and can become antigen-presenting cells. After the medulla, lymph exits the lymph node through the efferent vessel, out of the hilum, and continues on its path through other nodes. If you study this more extensively, you will see there is a whole lot more to this filtration process. But to recap, the lymph is delivered to the node from the afferent vessels, moves through the cortex and medulla, and is exposed to B cells, T cells, and macrophages. These cells will elicit an immune response if necessary, and the lymph finally exits through the efferent vessel. This process will repeat until the lymph makes its way back to the right or left subclavian vein. Another organ you will look at under the microscope is the spleen. It produces red blood cells in juveniles and has two areas you will need to identify. The first is called red pulp. This is generally a red blood cell graveyard. Red blood cells live about 120 days, and just like we get less flexible as we age, so do red blood cells. As they bend and flex to squeeze through the sinusoids of the spleen, the cell membranes rupture and the cellular components get recycled. We talked a little bit about this in Lab 2. In the second region, the white pulp, which appears purple due to the staining, contains white blood cells, specifically lymphocytes and macrophages. Again, these cells are monitoring for foreign antigens. Some people with color blindness may have a difficult time with this slide, so if you find it hard to see, let your TA know so he or she can make accommodations for you. Again, in this slide, you can see the concentrated areas where white blood cells populate the white pulp and the red pulp where blood filtration occurs. The third organ is the thymus. As you'll soon see, the thymus is divided into lobules, each with a cortex and medulla. These lobules are populated by T cells, and T cells mature in the thymus. While doing so, they gain self-recognition, meaning they learn to not destroy your own body, and they gain immunocompetence, meaning they learn how to be immune cells. Before we are even born and into early adolescence, the thymus is extremely active and larger to help our immune system develop. As we age, the thymus atrophies because our immune system is more mature and more developed. It was once thought that an enlarged thymus may have been a cause of sudden infant death syndrome, or SIDS, but that was later disproven. In this view, you can see the lobules of the thymus. Each of these lobules has a dark staining cortex and a lighter staining medulla. The cortex is where the T cells develop and train to become immunocompetent, and the medulla is where the now immunocompetent T cells replicate. The last lymphatic organs we will look at are the tonsils. We have 6 pairs that act as the first line of defense, trapping foreign material coming in through our noses and mouths. The first set is the palatine tonsils on the sides of the oropharynx near the soft palate. The lingual tonsils are at the back of the tongue, and the pharyngeal tonsils are at the back of the nasopharynx. Be sure to study this PowerPoint slide and the half-head models in lab for your test. Here are 2 different views of the tonsils under a microscope. You can see the tonsillar crypts, which are highly complicated networks of canals with special types of epithelial tissue and structures that can elicit immune responses. Here, you can see many lymphatic nodules, not lymph nodes, that line those canals. Sometimes debris trapped in the tonsillar crypts can get calcified and lodged in those crypts. This can lead to tonsil stones and can also cause bad breath. They usually form in the palatine tonsils. I definitely recommend checking out the hyperlink to a video showing tonsil stones getting removed. Many problems can occur in the lymphatic system, and those problems can lead to various medical conditions. Some pathologies associated with the lymph organs are tonsillitis or tonsil infections. In the image to the right, you can see signs of a bacterial infection of the palatine tonsils. Splenomegaly, or enlargement of the spleen. One of the most common causes is infectious mononucleosis, which we learned about back in Lab 2, and the Epstein-Barr virus. Thymus cancers, which you can see on this CT scan by the yellow arrow. Lymphomas, a group of blood cancers which affects the lymphocytes. Or there can be complications within the lymphatic vessels and the low pressure inside. Oftentimes there are obstructions to the flow. Remember at the beginning of the lecture I mentioned that if the flow is blocked or the pressure in the lymph vessels rise, it can lead to swelling? Well, one disease, lymphatic filariasis, is caused by mosquito-borne nematodes. If you remember your taxonomy from Bio 182, nematodes are roundworms, and you probably discussed this condition briefly in lab. The parasitic worms infiltrate the human lymphatic system and damage or block the lymphatic vessels, which leads to severe edema due to the increased pressure in the lymphatic capillaries, as well as a reduced uptake of interstitial fluid. The people in this image most likely have blockages in their inguinal and popliteal lymph nodes, which is causing severe edema in their thighs, legs, and feet. And last is lymphedema. This swelling can be hereditary or due to destruction or damage to the lymphatic system as a result of surgery or cancer therapy. This image shows different stages used by physicians to identify the severity of the swelling. So that's it for this lecture. Make sure you check out the slides in your free time in labs or during open hours before you take the online test. The slide boxes will be out every week for you to observe them. Be sure to make your drawings in the correct location of your notebooks and always refer to the syllabus for due dates. Thanks and have a great week.